Thank you for joining Cancer and the Nature of Life, our monthly series with some of the finest researchers in their field in the world. The series is part of the outreach for the NCI-funded Arizona Cancer Evolution Center, which we call ACE, and I'm Pauline Davies. Before we start today, put in your calendar the 17th of June when we'll have a wonderful discussion called What Animals Can Teach Us About Cancer. And we'll have Beata Uvari and Josh Schiffman, and that will be at 1 p.m. Pacific time. So an hour later than today, and that's because Beata is in Melbourne, Australia, and it will be very early in the morning for her. But right now we're talking about transmissible cancers in mammals, cancer cells that can pass directly from one animal to another. And I'm delighted to welcome first Elizabeth Murchison. Elizabeth is Professor of Comparative Oncology and Genetics at the University of Cambridge in the UK. She's in the Department of Veterinary Medicine and leads a team studying transmissible cancers in dogs and Tasmanian devils. And I think I'm correct in saying that it was Liz who discovered that the cancer raging through the population of devils in Tasmania wasn't caused by a virus, but was truly an infectious cancer. Liz will tell us more. And with her today is Amy Boddy from the University of California, Santa Barbara. Amy is a researcher with ACE and applies comparative biology and genomics to evolutionary theory and ecology with the ultimate goal of impacting human health and diseases, including cancer. So Liz will speak first, followed by Amy, and then we'll go to questions. And please use the Q&A for your questions, and that will help us keep track of them, and use the chat as you like. So turning over to you, Liz.
Okay, let me just share my screen. There we are. Well, thank you very much, Pauline, for that lovely welcome, and thank you everybody for coming. It's a really delight and pleasure to be able to join you today and to tell you a little bit about transmissible cancers in mammals. Including in Tasmanian devils. So some of you might have attended, I think, the brilliant lecture by Michael Metzger a few weeks ago, where he talked about transmissible cancers in bivalve mollusks. I'm going to pick up on that thread, but this time tell you about how transmissible cancers have occurred in mammals. Right, so let's start by just showing this very basic picture. I think all of us here are probably very aware of the fact that cancer is an evolutionary process which occurs when a normal cell, shown here, becomes a cancer cell, shown here, when it acquires a specific set of mutations which cause that cell to enter a state of proliferative autonomy. So each cancer could be considered its own unique evolutionary process whereby individual cells gain advantage and are selected for. And we can see this occurring here. So here's a cell in this cancer which has been selected for and gained advantage. However, the important point that I wanted to make here is that usually cancer is a very self-limiting evolutionary process because its continued survival is completely dependent on the body of its host. And so when the host dies, the cancer dies as well. So cancer is usually an evolutionary dead end. We work on cancers where this is not the case, the exceptional cases of transmissible cancers, cancers which are able to survive beyond the death of the original host that spawned them by the physical transplantation of living cancer cells between hosts. And we can see this happening here, actual living cancer cells being physically transplanted from one host to the next, allowing the continued survival of the same cancer lineage even long after the death of the original individual that first spawned the cancer in the first place. So fortunately, this type of disease is quite rare in nature. In fact, we only know of 13 examples in the whole of nature, only 3 of which occur in mammals. And I think you might have heard about the bivalves from Michael. So today I'm going to tell you about what we know about the 3 mammalian transmissible cancers. And I'm going to start by talking about Tasmanian devils, and in particular tell you about the really surprising discovery that we made a few years ago, that there are not 1 but 2 transmissible cancers in this species. And I'll also tell you about the canine transmissible cancer, which is an extraordinary cancer which is several thousand years old. Right, so let's start by talking about Tasmanian devils. So First of all, what is a Tasmanian devil? You in the United States, I think, are probably most familiar with the cartoon character Taz. Real Tasmanian devils, I think, are much cuter than the cartoon character. There's not really that much similarity, I would say. They are about the size of a small dog. They're well known for their terrifying screaming sound, which I'll play for you in a minute. And they're nocturnal scavengers. They're only found on the island of Tasmania, which is this island just down here to the south of mainland Australia, which is also where I grew up. It's a very beautiful place. And being marsupial mammals, Tasmanian devils give birth to tiny underdeveloped young. These are actually 4 Tasmanian devil babies as they look when they're actually born. So they're absolutely tiny. They crawl into the pouch and they complete their development in the pouch. And these are some young in the pouch about a few weeks of age. When we think of pouches, we normally think of kangaroos with that pocket-like upward-facing pouch. Tasmanian devils have a downward-facing pouch, which is essentially a fold in the belly skin. And at some stage, the young get too big to stay in the pouch. They start to fall out, like you can see here. And then the mother builds a den where she deposits the young, and she goes out hunting and scavenging by night and comes back to feed the young by day. And at about 7 or 8 months of age, the young become independent. They leave the mother, and after that they become very solitary animals. They don't tend to interact with each other very much. When they do interact, they tend to behave a bit like this. They tend to be quite aggressive, particularly around mating season. I just wanted to play this short video. I hope you'll be able to hear the sound, just to give you a sense of how they sound and how they tend to behave with each other. So I think that might give you a little bit of insight into how aggressive they can be. And believe it or not, these two are actually friends who live together in the same enclosure. So you can imagine how they would be if they really don't like each other. So I just wanted to also give you a little bit of insight into the field work that we do to collect devil samples. This is one of my PhD students a few years ago. I tried to take my group to Tasmania to do some field work. And it all starts actually with getting some bait. And sadly in Tasmania, there's a lot of roadkill, so it's not difficult to find some wallaby carcasses to use as bait. And what we do is to put the bait into traps, which are basically a piece of piping with a piece of with a trapdoor here. And the piece of bait is strung up down here. And when the animal goes in and pulls down on the bait, that closes the trapdoor there. So we bait these traps and we put them out in the landscape. And then we come back the next morning to see if there's anything in the trap. So if there's a closed trap, that's quite exciting. It's not necessarily a devil in there though. Actually, the second most common species to find is this one. It's a spotted tail quoll. Really beautiful animals, also marsupials. Sometimes we find really, really unexpected species. So for instance, once we trapped this little Jack Russell Terrier, which was quite a surprise. He was very, very happy to come out of the trap, but actually during the time we baited the trap and prepared it again, as soon as we left, he went straight back into the trap again. So wasn't the cleverest of little dogs. But actually most of the time when we do find a closed trap, we do find a devil inside, and usually they're curled up. And actually, unlike the way they behave with other devils, with humans they tend to be very docile. They get this torpor response when they're really scared, and actually they will usually just lie there. And what we can do is put them into a hessian sack, and then if we're very, very quiet, we can actually handle the devils and have a look for tumours, even collect samples if there are tumours present. And then the devils get released back into the wild again. And this is actually just a little juvenile Tasmanian devil that we trapped a few years ago, which I put there just to just to illustrate how unique and also just adorable this species is. They're not just ferocious, angry animals, they're also just absolutely unique and wonderful. And sadly, this species is under threat due to this transmissible cancer. So I'll tell you a bit about that. So the story of this transmissible cancer started with this one photograph of a devil with a facial tumour taken in 1996. So at the time, it was just thought to be a one-off case with a devil with an unusual cancer. But actually, we now know that this is the first known sighting of infectious disease affecting this population. So this photograph was taken in 1996 in northeastern Tasmania. And in subsequent years, there were more and more sightings of devils with facial tumours. But it wasn't until the early to mid 2000s that it became clear that this is a new type of infectious disease spreading through the devil population. And by this stage, it was really too late to try to stop the disease in its tracks. It's continued to spread. And now, in 2022, the picture's a bit like this. The only place which is confirmed to be disease-free is this area in far northwestern Tasmania. And it's thought to be just a matter of time before the disease arrives there. So what is this disease? Oh, I should also mention that this is causing massive declines, which I'll talk a little bit more in a minute. So what is this disease? Well, the disease was named devil facial tumour 1 or DFT1 and described as a disease that leads to the appearance of tumours on the face or inside the mouth of affected animals. This is a very typical case here. The tumours usually start as small lesions and then rapidly progress into these large masses. If we were to look cytologically at the cells of these tumours, we would find that these tumours are composed of these small, round, highly undifferentiated cells. And if we were to take a tissue biopsy and look at the tissue in these tumours, we'd find very densely cellular bundles like this. So the blue staining is showing the tumour cells, which are positive for a particular marker called PRX, where we can see the brown staining here. So actually, the first evidence that this cancer really was spread by the transfer of cancer cells and not a virus, which was what was originally thought, was not my work. It was actually work from two colleagues, Anne-Marie Pierce and Kate Swift, who were cytogeneticists studying Tasmanian devil chromosomes. And Tasmanian devils have fairly simple chromosomes. They only have 7 pairs of chromosomes shown here, including the sex chromosomes. DFT1 has this highly rearranged set of chromosomes. So you can see chromosome 2 appears to be missing altogether, as do the sex chromosomes, and we have these rearranged chromosomes here. So this itself is not surprising because aneuploidy, or rearrangement in the chromosomes, is really common in cancer. But what was really surprising and really striking was that all the devil tumours which were examined, I'm just showing 4 here, had exactly the same changes in the chromosomes. And we can see in particular this long, unusual looking chromosome here. Now in cancer, each cancer usually has its own unique changes in its chromosomes. And the fact that all of these tumours shared not just similar, but actually almost identical changes in their chromosomes suggested that these indeed actually were the same cancer, which first arose in one devil and then spread through the population by the transfer of cancer cells. And how would it be spread? Well, that's where the biting behavior becomes important. So we think that when devils bite each other, they actually physically transplant tumor cells from one animal to the next, which are somehow able to escape the immune system, which I'll touch on in a minute. So I wanted to actually, the way I got involved was to try to confirm this using genetics. So basically by testing whether the DNA from the devils themselves and from their tumours was genetically unmatched, because if it's a transmissible cancer, then we would expect the DNA of the tumour and the host to be different from one another. So we put this to the test using what's called microsatellites, which are genetic markers, which are basically simple repeats in the genome. We all have them in our genomes, including devils. They don't really do anything much most of the time, but they're quite useful because they change quite frequently. And so they're useful for identifying individuals. And so what we were able to do was to establish a panel of microsatellites. And the numbers here just represent the different alleles, and we don't need to worry too much about that. But what I wanted to show you was some results. So basically, this is the genotype. This is showing the genome of one particular normal devil. And what we can see is that he's got 2 copies of every chromosome, and at this particular position here, he's got 1 copy of the blue one and 1 copy of the red one. And here he's got one copy of the blue one and one copy of the grey one. Here he's got two blue copies. He's got one blue, one red, etc. So he's got his own unique fingerprint, is what I want to say. So what happened when we looked at these DFT1 tumours? Well, indeed, we found that all of these tumours were identical across all of these genetic markers, indicating that they actually are the same, belong to the same individual. And furthermore, and importantly, in each case, the tumour was genetically unmatched with its host. So this is a tumour which was taken from this host. This is a tumour taken from this host. And you can see in each case, they're genetically different. So this really does confirm that these tumours did not arise from the cells of their own host, as most cancers do, but rather they arose in one original devil, which we happen to know was a female, actually. This devil, one cell in this devil became cancerous, grew into a tumour. Rather than dying together with that host, as most cancers do, it spread to another devil, or perhaps to several more devils, and from there it spread all around Tasmania. So when we look at these cells, these DFT1 cancer cells under the microscope, we're looking at something extremely unique. So this is not just a cancer, it's a cancer which has become an infectious disease, which is able to escape the immune system despite being a foreign graft. And actually it's the only cancer that we know of in nature which is threatening the existence of the species. So how does this cancer escape the immune system? We still don't fully understand this, but a really important part of this is was elucidated by colleague Hannah Siddle a few years ago. So what Hannah found was that DFT1 cells actually don't express a set of molecules known as MHC class I molecules, which are normally expressed on all nucleated cells, and which are the main flag that the immune system sees to detect self and non-self. So this is actually what needs to be matched when you do organ grafts, organ transplants between people, because MHC is what the immune system targets. So if there's no MHC class I at all, then the immune system doesn't really have a lot to see and can't really tell that the cell doesn't belong to itself. So that's a really important part of how this works, but not the complete story. So what's the outlook for Tasmanian devils? Well, at the moment it's looking pretty grim. This is a plot showing the relative devil population size as a function of years since DFT1 arrival. And you can see that there's this catastrophic drop in devil numbers. And it's unclear what the future holds. And so to prepare for the worst, the Tasmanian government has established an insurance program, which is essentially a captive breeding population of Tasmanian devils, making sure that Tasmanian devils and their genetic diversity are maintained in large enclosures and even an island off the coast of Tasmania, which has been made into a devil sanctuary, so that if the species really does go extinct in the wild, then they could be reintroduced. So this is all looking pretty grim, and we in the devil community, we're all working on this and trying to find the best possible solutions to help the devils. And then something extremely unsettling and unexpected happened, and that is that we discovered that there was actually a second transmissible cancer in this species, and this is something completely unexpected. So this all started with this very sad case of this devil with this horrific tumour. I'm not going to dwell on that picture. So that devil called RV was actually found down here in southeastern Tasmania in 2014. As you can see, his tumour, very horrific tumour, was very suggestive of DFT1. But when my colleague Ruth Pye took some samples from that tumour to do some basic diagnostics, some cytogenetic diagnostics, she found that his chromosomes actually didn't look anything like DFT1. Remember how DFT1 was missing chromosome 2, missing chromosome, the sex chromosomes? RV's tumour looked pretty much like a normal male. And indeed, you can even see the X and Y sex chromosomes here. The only difference from a normal male was missing chromosome 6 and a bit of extra material here on chromosome 2. So this was really bizarre and seemed to suggest that RV just had a tumor, which was a normal tumor, which arose from his own cells, which just happened to look a lot like DFT1. So that's what we thought. And in fact, also histologically, his tumour looked different. It didn't have these bundle structures. That was what we thought until a few months later, early 2015, we found this other devil 15 kilometers from where RV had been found, again with these absolutely horrific tumours, very, very typical of DFT1. But again, when Ruth looked at the chromosomes of this second tumour, she found, to her absolute surprise, that the chromosomes again didn't look anything like DFT1, but really rather looked identical to RV. And in fact, you can see the missing chromosome 6 and the extra long chromosome 2 and the X and Y sex chromosomes. Histologically as well, looking at the tissues, SN's tumour looked really just like RV's tumour. So this really seemed to suggest that actually these two devils had a transmissible cancer which was different to DFT1. And so we wanted to test this time using genetics. So they sent some samples over to us and we used the same panel of markers to look at this. And so the first thing we did was to look at RV and SN. And as you can see, the tumours are genetically identical across all of these loci. So this indeed confirms that these belong to a transmissible cancer. And furthermore, they were genetically distinct from their hosts. So they did not arise from their hosts. But then the most crazy moment, I think this is the most surprising moment in my entire scientific career, was to find that RV and SN's tumours were completely genetically distinct from those of DFT1, really suggesting that this is a different transmissible cancer. Doesn't totally prove it at this stage because, of course, these markers can also mutate in tumours, and so it's possible that these could have originally come from the same cancer but then mutated and become different. But we were actually able to confirm or to rule out that possibility because of the presence of the Y chromosome. So as I mentioned before, we happen to know that DFT1, the first cancer, came from a female devil, and given that there's a Y chromosome here, that really shows that this cancer must have come from a male. So we now know that there are actually two transmissible cancers, and the second one we've named devil facial tumor 2, or DFT2. It's also spread by biting. It actually grossly looks completely indistinguishable from DFT1. And so far, it's only been found in this small peninsula in southeastern Tasmania. But really bizarrely, and this is something we still don't understand, is that DFT2 cells actually do express that important marker MHC class 1 on their cell surface. So how it is that they escape the immune system is still unclear, but the main hypothesis, which has been proposed by my colleague Hannah Siddle, is that perhaps there's too little genetic diversity or diversity in MHC within this population in this peninsula in southeast Tasmania to allow hosts to detect this as foreign. So we know of two different transmissible cancers in Tasmanian devils, one coming from a female and one coming from a male. So what does this really mean for Tasmanian devils? This was really mind-blowing. I mean, we really didn't expect this. Because previously we thought that Tasmanian devils were just really unlucky to get a transmissible cancer. But now that we know that there are two transmissible cancers in this species, it seems to suggest that actually this species holds a very particular vulnerability to developing this type of disease. And what could that be? Well, to me, it seems likely that it's partly to do with their biting behavior, which gives an opportunity for the disease to spread from one host to another. And maybe coupled with that, also their relatively low genetic diversity, which comes from previous bottlenecks that they've gone through and living on a fairly small island. So that means that it's possibly less likely that that the immune system would be able to detect a foreign cancer as effectively. But also this raises the question, how is it that these cancers have arisen now and we've never seen these before in Tasmanian devils? Well, I think it's very likely that probably these cancers have occurred in the past but have just escaped detection. I think it's also probably quite likely that they will occur again in the future. So I think probably from the conservation perspective, we have to watch out for these things happening again in Tasmanian devils. So coming back to this picture, I just now very briefly in the last few minutes wanted to introduce you to the canine transmissible cancer, which is really, really fascinating. So to introduce you to this, I wanted to introduce you to this dog here, Kimbo. He's a dog who was adopted by a family in the Mombasa region of Kenya a few years ago. And they noticed some blood trickling from his genital region shortly after they adopted him. They took him to the vet, who happened to be one of our collaborators actually. And the vet examined Kimbo and discovered this horrible tumour at the base of Kimbo's penis. So the owners were shocked and horrified, but the vet was not at all, because he was very familiar with this cancer. It's one of the most common cancers in dogs in Kenya, and indeed in most parts of the world. It's the canine transmissible venereal tumour. It's a sexually transmitted cancer which affects dogs. It causes tumours on the external genitalia in both males and females. These tumours are very different in the way they behave from the devil tumours. They tend to be very localized. They just kind of sit on the surface of the tissue most of the time, and they don't tend to metastasize very often. And they're also highly curable. With a simple course of chemotherapy, most dogs will completely recover from this disease. So this is a transmissible cancer which first arose also in a single dog and spreads through the population by the transfer of living cancer cells by mating. And actually this cancer is now found all over the world. Each dot on the map here is a location where we've got evidence that this cancer, CTVT, is actually endemic in dogs. It's really present in most parts of the world, and actually its presence is predicted by the presence of free-roaming dog populations. So that's why we don't tend to see it a lot in parts of North America or Northern Europe or parts of Australia. It's because we require a free-roaming sexually active host population to sustain this cancer. But this cancer is really found almost everywhere, which is extraordinary when you think that this is just one single cancer. So that means that it might be quite old to have spread so far around the world, but exactly how old? To test this, we wanted to use mutations, and particularly to use mutations as a kind of molecular clock to find out how long ago this cancer first arose. So some mutations arise at a constant rate over time. Some mutations arise due to kind of sporadic exposures to things like UV light from the sun, but others just arise at a constant rate over time. And one type of mutation which has this characteristic is Cs when they occur right next to Gs. And those Cs occurring right next to Gs in the DNA sequence are very prone to getting a very particular type of mutation, which happens constantly over time. It's kind of like a ticking clock. The only thing is that the rate at which that clock ticks varies from tissue type to tissue type and from species to species. So in order to use this to estimate the age of CTVT, we first needed to estimate the rate at which it ticked in CTVT. So we did some analyses to estimate that using direct transmission events when we knew that dog A had transmitted to dog B, and we estimated that there are about 43 of these mutations per year. And so just by counting the number of these mutations in CTVT, we were then able to extrapolate and to find that CTVT probably arose about 6,000 years ago. So there's a huge confidence interval on this. We think it could be somewhere, it could be anywhere around that, but we think probably a few thousand years is realistic. So when we look at this cancer here, we're actually looking at a cancer which first arose in a dog that lived 6,000 years ago or so. It's just extraordinary. We wanted to find out a little bit more about this animal. So actually, this animal's genome is still encapsulated within these cells that it spawned. And so by looking at the genetics of these cells, we're able to say something about the identity of that dog that first gave rise to the cancer. So we first of all made a family tree of dogs and looked to see where CTVT fit into that. So this is a family tree. We've got some wild canids here, coyotes and wolves. And then if you look at the modern dogs, there's northern breed dogs, East Asian dogs, and West Eurasian dogs. Where does CTVT fit in here? Well, actually, it's a sister group to these Arctic sled dogs. So we think that the dog that gave rise to CTVT was some kind of a husky or Malamute type of dog, a sled dog. We wanted to go a little bit further because actually quite a lot is known now about the genetics of dog morphology and coat color and even behavior. And so what we were able to do was go into the genome of CTVT, look at the particular loci which confer various traits, and read out what the likely phenotype of or characteristic of this founder dog was. So for instance, this gene here on chromosome 24, its sequence defines the coat color of the dog, and we found that probably the original dog had an agouti or a wolfy type of coat color. We also figured out that it probably had short, straight or wavy hair, pointy ears, was probably of medium to large size, and even some of its behavioral characteristics. For instance, we found that it probably had no increased risk of obsessive compulsive disorder. And actually we were able to draw a picture of this dog. So kind of bring it back to life based on the genetics, its genetic legacy and the cancer that it spawned, which is a really amazing concept, I think. So these cells are really pretty extraordinary, and by looking at them, we're stretching back thousands of years of history. How does CTVT escape the immune system? Well, it's the same, similar to DFT1, it appears that these cells have lost MHC class I expression. So again, the immune system doesn't have its major target to see on the cell surface, although there's still much more to be learned there. So coming back to this picture to finish up, I would like to say that although we've been finding more transmissible cancers in recent times, I would say transmissible cancers are still very rare in nature. We only know of 13 of them in entire nature. So it looks like transmissible cancers are unlikely to occur. And we know that normal cancers which stay in their hosts are pretty common. So why is it that, that becoming transmissible is so unlikely? I'd say it's probably because two quite rare things have to happen in order for a cancer to become transmissible. First of all, there has to be a stable transmission route, like biting in the case of devils or sexual transmission in the case of dogs. And that might be quite unlikely to be present. And secondly, the cancer cells have to have a mechanism of escaping the immune system. So they have to, most cases, have downregulated MHC class I and possibly other things as well. And so having those two circumstances, which are probably both in themselves fairly rare, occurring together is probably very unlikely. However, we know actually it's become increasingly apparent that once a niche exists in nature, so for instance, in Tasmanian devils or indeed in bivalve mollusks, then these cancers can occur repeatedly, can arise repeatedly. And once they arise, they can really spread widely through species and become a huge problem for species, even species, endangering species survival. And really broadly, I would say they also very strongly illustrate the potential of cancer cells to be malignant and invasive parasites in their own right. Right, I'm going to finish there and just quickly acknowledge my group. Here we are on our lab hunting trip and our funding mostly from Wellcome, and finish up. And thank you very much. Happy to take questions.
Gosh, that was absolutely fantastic, Elizabeth. And I've certainly thought of a few questions, but first of all, let's turn to Amy. So Amy, do you want to comment on Elizabeth's talk?
Yeah, wonderful talk. I'm always in awe when I see you speak about these transmissible cancers. And I can't help but think, and there's two things I want to target before we open up for questions. And one of them is looking at cancer in zoo populations in Tasmanian devils. And we see quite high cancer there too, even though it's not facial tumors. And I think that's just something we could discuss that might be inherent about their biology, and it might, you know, be something of this genetic bottleneck. But the other thing that I think would be really interesting to chat about a little bit is, I couldn't help but think when you're defining you need a stable transmission route and you have to escape the immune system. And so I've been really interested in pregnancy in mammals. and there is this cellular exchange that happens, right? And so another area of research I study is microchimerism. And this is the transfer of, well, microchimerism by definition is harboring a few cells from a genetically different host. I don't know if we'd actually define these transmissible cancers as microchimerism. It seems like it'd be more like chimerism, macrochimerism, or just chimerism, right? But it's inherently normal in mammalian biology to have these swapping of cells that are only half related to you, and you have a stable transmission, and you can escape the immune system because we see these cells persisting in the lifetime of their host. And it makes me think that mammals might be more vulnerable. I know bivalves are a different species that are not mammals that also have transmissible cancer. But
Amy, I'll just point out for people who are not biologists by training. So you're talking about pregnancy when you know, some of the cells from the child, the babies, is staying in the mother. Is that right?
Yes,
okay.
Exactly. And that's actually, and there are a few cases in humans. So there's a route for a transfer of cells, and it seems to happen in every pregnancy. And then these cells last for a lifetime in either the offspring or the mother's body. So we're all a little bit microchimeric, all right? But there's also certain cases, I looked it up just last night, there's about 50 cases reported in the literature of transmissible cancer in humans through this route, again, during pregnancy. And it seems, again, to be very rare. It's not that rare to be diagnosed with cancer while a human woman is pregnant, but it's actually rare for it to transfer. And so I wonder if there's more.
Amy, again, which way does it transfer from the fetal tissue to the mother?
I love this question. Yeah, thank you, Pauline. So all instances, it's the maternal body having cancer and transmitting it to the fetus. Mm-hmm. Most of them get lodged in the placenta, but there are cases where they go on and metastasize to the fetus. There's no evidence of fetal cells going in cancerous fetal cells and metastasizing to the maternal host, which is interesting because when we're looking at microchimerism, there's more fetal cells that get transferred during pregnancy. So,
but and also I think there's some cases of it being transmitted during childbirth. So there's been some cases of cervical cancer being transmitted during childbirth to the offspring.
Yeah, some cervical cancers, I believe, were transferred that way. And so when you're half genetically identical to the host, then again, there's this immune escape. But it just makes me question, why isn't this, like, what are our mechanisms of defense? Why aren't we seeing it so much more often than we actually do? There's only 50 case reports and 200 years of observing it. There's been millions and billions of births, right?
Yeah, well, I guess first of all, those types of cancer transmissions from for instance, from mother to fetus, or also there's some cases in the literature, for instance, of surgical accidents or lab accidents where cancer accidentally gets transmitted from one person to another, or also actually organ donation. There are some cases where there was an occult cancer which wasn't really known when an organ donor was screened, and it was transmitted to a recipient and then developed into a full-blown cancer in a recipient. But all these cases, I think, they only involve small clusters of individuals, and there's no stable transmission route like we see in these cases in Tasmanian dogs and bivalve mollusks. And so yes, they're the same concept, but they're by definition quite limited because there's no transmission route beyond those two or small number of individuals within the transmission cluster. So, I wonder if in humans there would be any potential for that to arise, maybe starting out using one of these routes and then spreading further. As you say, I mean, rare events occur and there are billions of humans. So I think it is possible that a transmissible cancer could arise with a stable transmission route, could arise in humans. To me, it looks like sexual transmission would probably be the most likely mechanism, since we've seen that already in dogs and since that would be possible in humans. But it seems to me that if that happened, it probably would be fairly easy to detect and probably bring under control before it became really out of control. So I think it's something that maybe everybody should be aware of, particularly clinicians, that it would be a possibility, but I don't think everybody should be really worried about it as a possibility. It's really unlikely to occur. And even if it did, I think it would be easily controllable.
Yeah. Do you think, can I have a follow-up question? Do you think there's something inherently about the stability of these tumors that is important too? Like a lot of tumors are pretty, like you mentioned, unstable, like you have tons of mutations, but I mean, 6,000 years with the same karyotype, is that unique? Like, would you predict that? Or have they hit this evolutionary sweet spot in terms of, like, I don't know, genome chaos and things like that? Yeah.
Yeah, it's a really interesting question. This dog cancer, CTVT, is extremely stable, at least at the level of its karyotype. So it looks like it's probably got pretty good DNA repair mechanisms intact, to which many cancers lose their DNA repair mechanisms, and that might be increasing their evolutionary potential to adapt by creating variation. But in this dog cancer, probably maintaining some level of genome integrity has probably been key for its long-term survival. So it is a really unusual cancer from that perspective. And even just the existence of these cancers does make you question kind of what is it that's, which features are important for cancer evolution in the long term? And are there differences between kind of very short term, like we generally see in humans and other animals, tumors which just stay in the same host, and which can give cancers an ability to survive over long time periods.
So I wonder if we should just go to one or two questions, and
let's do it.
Amy, chip in whenever. So Paul Davies has asked something that has been on my mind for forever. How come Tasmanian devils still exist? We've had 2 cases of cancer originating in recent decades. If 2 has happened, couldn't more cases have happened? And if so, how have the Tasmanian devils survived?
And this also ties in with Amy's first point about the fact that they are also very prone to cancer of the non-transmissible type. So they do seem to be a very vulnerable species. And all I can say is that it seems possible to me that these transmissible cancers in the past, they might have arisen and maybe died out also in localized devil populations and not spread as widely and threatened the population as much as this first one we've seen in recent times. And I actually think it's quite possible that humans have modified the environment in such a way that devil ecology has been quite transformed, and maybe leading to greater chance that if these diseases arise, that they spread. So for instance, by building roads, that gives devils a route to move around through the environment. And agriculture has artificially inflated devil populations in the 20th century. So possibly in the past, devil populations might have remained more separate. These diseases might not have had the chance to spread as much. Also, maybe there were some chance events in this particular case. Maybe it got off to a running start and therefore spread faster and more than others in the past. So that's all I can really say, that I think it's very likely these have happened before, but just they've survived it. So maybe that does give us hope about this one as well.
I hope so. Do you think they might, some of them might start developing some immunity to the cancer cells in some way?
Yeah, potentially. It's actually been suggested that that might be happening. And if that were to happen, then that would be really wonderful news. And we have actually seen in the wild evidence of devils which have established tumours and these spontaneously regress, that has been seen from time to time. We don't know how common that is, it's probably quite rare. But that means that there are devils out there which are able to fight off this disease. Whether that's genetic or not, we don't really know for sure, but if it is, then yes, there's a lot of cause for hope.
Yeah. Can I build off that? And I noticed Carlo Mealy has some immune questions in the chat. There's a question on, there are many other mechanisms of immune escape other than suppressing MHC, and have they been looked at, especially for DFT2? If I could build off that, there's more than one MHC loci, right? And has that been looked at as well?
Yeah, great questions. So those of you who know a little bit more about immunology probably know that it's much more complex than just MHC. So actually cells which lose expression of MHC normally become targeted by a group of cells known as natural killer cells. And part of natural killer cells' job is to find and kill cells which are not expressing MHC because they're abnormal. And that is somehow not happening in any of these cases. And we believe that there must be some other mechanisms occurring locally, establishing immunosuppressive microenvironments in these tumours. But we still don't know the mechanisms of that in any of these cancers in great detail. The case of DFT2 is really interesting that it actually is expressing MHC. It's just so surprising. And yeah, I think it's likely that there's maybe that cancer is just within a population that's so closely related to the founder devil. And actually Hannah Siddle has found that the MHC molecules which are expressed are very commonly expressed in devils in that area. So that leads to the hypothesis that maybe in order to spread further, maybe that we'll see that cancer losing MHC expression. But so far, that hasn't really been very well established. In terms of MHC, yeah, so there are a number of MHC genes, but actually most of these cancers, what they seem to do is shut off one key common gene known as beta-2 microglobulin, which all MHC genes require in order to be displayed. And so by focusing that kind of node, targeting that node, then MHC gets shut down altogether.
We've got some more questions coming through. We've got a question from Steffi. How much do we know about the transmissible cancer cells possibly controlling the aggressive behavior of the Tasmanian devils? Does it have any influence on their behavior, the cancer cells?
Well, yeah, great question, Steffi. That might be something that you might expect from an evolutionary perspective that might be an advantage for devil cells. If they were to make their hosts more aggressive, then maybe that could lead to more chances for transmission. That's not something that we've got any evidence for at the moment, but it would be really interesting to study that.
Okay, and from Michael Metzger, is there any bias to DFT2 to affect males more than females since it's a male cancer?
Oh, brilliant question,
yeah,
Michael.
Brilliant, Michael.
Yeah, I was hoping somebody would ask that. Yes, so DFT2 is very strongly skewed towards male hosts. In fact, more than 75% of the early cohort was found in males. And so of course it leads to the question as to whether Y chromosome antigens are being targeted by the immune system in females because they're completely foreign to the female body. And that's something that we've been looking at. And in fact, indeed, we found that in DFT2, there have been repeated losses in recent years of the Y chromosome from DFT2 cells, which would be indicative potentially or suggestive of selection for loss of the Y and loss of Y chromosome antigens and potentially more opportunities to grow in female hosts. And so what we're waiting for now is for enough devils with these Y lost DFT2 cells to see if the sex imbalance gets equalized again, and that will really prove it, I think.
And from Carlo, he's saying, there are many other mechanisms of immune escape other than suppressing.
Oh, we already We already
Oh,
asked
I've done that one, sorry.
Yeah, but the second one about if they're Schwann cells.
Yeah, Schwann cells. I see that.
Are both DFT1 and DFT2 both Schwann cell tumors? That seems surprising if vulnerability was just about biting and genetic homogeneity. That's usually a rare cancer.
Yeah, so yes, I think that's another complexity. Both of these cancers derive from the same cell type, Schwann cells, which is the type of cell which normally protects and insulates peripheral nerves. It's a very rare type of cancer in general in humans and in other species, particularly this aggressive form. So yeah, it does seem that it's that particular cell type which is poised to become transmissible in devils. And the reason for that is not entirely clear, but it's really interesting, the connection with biting here, because Schwann cells are usually fairly inert cells that don't really do that much other than just protect nerves. But when there's an injury, then they go into this very active state. It's called a repair Schwann cell, where they divide and they do all sorts of things to recruit repair cells and to repair the injury. And it appears in both cases, DFT1 and DFT2, the cancer may have arisen from some kind of repair Schwann cell stuck in the repair mode. So normally when the injury is repaired, then they go back to being mature, slightly inert cells. But it looks like in these cases, they've got stuck in this proliferative repair mode. And so that, of course, makes you think about devils and how they bite each other, and they probably get a lot of nerve injuries around their faces. So maybe the biting behavior also predisposes them to getting this kind of cancer in the first place.
So interesting. So the question from Paul is, what about their closest extant relatives, genetically speaking? Have you looked at them to see if there's anything that could predispose them to transmissible cancers? You know what the relatives are?
Well, there's a whole group of carnivorous marsupials, including the quolls that I showed you in the picture. So far, no transmissible cancers have been found in any of the other dasyurids or transmissible or carnivorous marsupials. So whether or not it's something specific to devils or whether other dasyurid marsupials might also be vulnerable, we don't know. But devils do have this very specific biting behavior, which is very likely an important part of it.
Can I build off of that? That's so interesting.
Yeah.
And Carlo Meli asked a question about if there's any evidence that infected devils are more bitey. And I want to add on this question in a second parter. Like, do you think that there could be a trade-off like this wound healing? If you're a bitey species, then you probably have to have these unique wound healings to always be repairing that. And what are your thoughts on that as well?
Yeah, great questions. And yes, actually, one thing that one colleague, Rodrigo Hamid, has found is that it's not just that this cancer in devils doesn't just go from the biter to the bitten. It can also go from the bitten to the biter. So if a devil bites a tumor, that seems to be another mechanism of tumor transmission. And so it's not necessarily the case that the most aggressive devils are those that are going to transmit the disease.
I see.
It can be a little bit counterintuitive that even those devils that are less likely to bite could be bitten or the other way around. So yeah, and so it would be really interesting to know whether the behavior of devils with tumors and without tumors differs, but we don't have clear evidence of that yet. The wound healing question is really interesting too, and it would be really interesting to know if devils are any different to any of its most closely related species in terms of wound healing genetic pathways, and also observations as to how well wounds are healed, and if there's any differences there that could be contributing.
It might even be worth looking at fossil bones of marsupials that have gone extinct, just to find out if any of those were susceptible to cancer, just looking at historical record as well.
We've actually done that with devils, really in a search for whether other transmissible cancers have occurred in the past, because DFT actually really does leave an impact on the bones quite frequently. But so far, there's been no evidence of anything like that. But yeah, maybe there are some fossil bones out there which could tell us that transmissible cancers occurred in the past in devils.
And Michael in the chat is saying, in terms of treatment possibilities for devils, has anyone tried to make a devil anti-PD-L1 to boost the non-self response?
Yes, actually, the vaccine group led by Andy Fleece in Tasmania, they are working on PD-L1 and have been having thoughts in these directions. So one thing that's really interesting is that actually in DFT1, you can induce DFT1 cells to express MHC class 1. Yeah, so I made a wrong point before. Actually, it's only in CTVT where beta-2M is targeted. In DFT1, actually, there's epigenetic downregulation of MHC protein genes. And if you treat these cells with a cytokine called interferon gamma, you can actually cause the cells to express MHC once again. But interestingly, alongside MHC, PD-L1 comes up as well. So it makes it challenging, because it's actually those cells which might be more immunogenic which are also expressing this anti-immunogenic molecule. But it's certainly a really interesting thing to explore.
Yeah, and then
interesting. Pauline, can I ask one real quick one following up on that?
Please do.
I just keep thinking, as you're mentioning this, about infectious disease, right? It's an infectious cancer. And so infectious diseases, you sometimes see this trade-off with virulence in transmission, right? Would you predict that you would eventually see this trade-off? It seems super transmissible and super virulent. Is there going to be like a tipping point here, or?
Yeah, great question. And so I guess that's getting to the idea that if you're a disease which is very virulent and kills the host really fast, then if there's a variant which coexists with its host for longer, it might have more transmission opportunities and therefore might be favored in the longer term. But I think that one thing that really puts a spanner in the works of that is that in devils, we've actually seen evidence that they could be co-infected with different types of tumors. So that means that it's probably less likely that that might happen, because if you had a co-infection situation, you could imagine that, you know, if you're the longer living type of tumor, the less virulent tumor, then it doesn't get the advantage because you've got co-infection with a more virulent form. So I don't know, it's very complex. It's difficult to predict what's going to happen, I would say.
Yeah.
So we've got a question from Anurag, and he's saying, any mathematical modeling being done in this realm, because he thinks it's ripe for some nice theory. What do you think?
Yes, there's quite a bit of work in that area from various groups. Yeah, and in terms of outcomes for devils, there have been a number of models which have made various different predictions. But of course, it's really difficult when you're working with very limited field information. So I think in the coming years, we're probably going to get really important data, which might allow us to predict more what's going to happen next.
Well, we've just coming up to the hour, and so I want to thank all of you who've come to listen to this, and we'll carry on if we've got some more questions, but I know a lot of you have to go. But before you do, just remember to tune in on the 17th of June to hear Beata Uvari and Josh Schiffman discuss what animals can teach us about cancer. And don't forget, if you want to see this discussion again, you can go to the Arizona Cancer Evolution Center and find it on Google, and we'll have the link up in a few days. But we'll carry on for now. But any of you who have to go, thanks very much for tuning in. But turning back to Amy and Elizabeth, I mean, it's been what a great discussion. This is something that can really take the imagination of people of all ages, can't it? Because you know, we want to save the species. I want to ask, what is the Australian government doing to help preserve this species? Because they must be surely desperately concerned as well.
Yes, so they are very much behind the insurance program, insurance population program, which involves keeping a big captive breeding population of devils, which is spread across both zoos in mainland Australia and in other parts of the world. And also there are large free range enclosures and even an island. That picture that I showed during the talk of the devil running off is actually the first devil being released onto an island which has been made into a devil sanctuary, which previously didn't have any devils on it. So these populations are very intensively managed to ensure that they retain genetic diversity and retain the genetic diversity of the species. And at the moment, that's the main thrust of the conservation program, to ensure that the species is maintained, at least in captivity. There's also a lot of work on developing a vaccine with colleagues in Tasmania, and that's been really challenging just because of the lack of antigens or lack of anything that's different between these cancer cells and the host cells, and they're trying to find these very specific differences and then target a vaccine towards that. But so far, unfortunately, there hasn't been very much success in developing a prophylactic vaccine.
Which really ties into the link between, human cancers, because it is a very difficult thing, isn't it, to distinguish between cancer cells and regular human tissue. Maybe, this could give a clue as to how to go about doing it in humans as well, which is, of course, what everyone wants, as well as to preserve all the animals.
Yeah, but in this case, we're in a better situation because it's the same cancer. So if we could find just something that worked, then all devils could be immunized that way. But of course, in humans, it would have to be patient specific.
Yeah.
Yes.
Well, carry on, Amy.
I just noticed one more question, and Alicia asked it, how old are these tumors? Have you estimated the origins when that devil, how long ago that, patient 0 lived?
Yes, we have actually, for both DFT1 and DFT2. And DFT1, it suggests that it arose in the mid-1980s, probably around 1986 or so. So about 10 years before it was first observed, which is quite a long gap. It was kind of longer than we had expected. So it meant that it kind of flew under the radar for quite some time, for a whole decade. And obviously there wasn't that much monitoring at that time, but there were some field projects going on and it wasn't observed. So it looks like it possibly stayed at fairly low prevalence for some of its first years. And maybe that's telling us something about the R number and how many secondary infections per infection, and maybe the generation time. So the time from getting infected to going on and infecting the next host might be a bit longer than we had kind of assumed.
Gosh, a lesson for all of us. Take note of what's happening in nature and look carefully. So I want to thank you both. It's been absolutely fascinating. Thank you so much, Elizabeth, and thank you so much, Amy. It's been a wonderful discussion, and I do hope we can talk to you again at some stage. Thank you.
It was a pleasure, and thank you so much for the invitation. It was wonderful to see you both again.
Oh, lovely.
Yes, wonderful talk as always. Yes.
Really, thank you so much. I'll close this now.